Great. Well, thank you to the organizers for the invitation. It's great to be back at IPAM. The last time I was at IPAM, I was just talking about was 2011, so it's been quite a long time. And that was a program in genomics, which turned out to be quite influential in my career, very early in my career. And in fact, it's so influential that it spawned a summer school, which still happens every summer at UCLA, called the Computational Genomics Summer Institute. So those of you that are more genomics inclined should check that out. And I hope for some of the younger faculty and trainees, maybe this meeting is similarly influential in your career. So thank you to IPAM for hosting us here. So I'm going to pick up on some of the themes as a tutorial. It's actually picking up some of the themes that Dominic described yesterday on cancer evolution, but from a slightly different perspective, more algorithmic perspective, more informed by some of the modern sequencing and genomics data. So I think we heard a lot yesterday, and we'll hear more about how cancer is an evolutionary process, driven largely, although not exclusively, by the accumulation of somatic mutations during the lifetime of the individual and leading to mutations that prescribe a selective advantage and clonal expansions and additional mutations, so that when we measure a tumor at the present time, it's a real heterogeneous mass. It's a mix of both normal cells that support or inhibit the tumor, cancerous cells with various somatic mutations that actually reflect this past evolutionary process. And so, like any evolutionary process, you can attempt to reconstruct the past from the present. And so try to measure these mutations and go back in time and build out the tree, the phylogenetic tree, that relates the present-day cells, or sometimes groups of cells or clones, depending on our measurement resolution, and their ancestor relationships back to the founder cell of the tumor. And this is a very helpful perspective for a variety of reasons. Tell us perhaps something about the early events of the tumor. If we do this across many patients, we might be able to see common patterns across their independent evolutionary events, independent in each patient, but of course correlated by the mutations and genes that are mutated. I'm going to talk a little bit about how we might use this to actually understand the process of metastases and how cells leave the primary tumor and seed tumors at other anatomical locations. And then clinically, and this has been challenging, but I think there's a lot of interesting work yet to be done in trying to figure out how to perhaps treat tumors in relation to either their past evolutionary history, the early events versus the late events, the heterogeneity within tumors, sort of correlations between the driver mutations that we see. And I think we heard a lot yesterday and maybe more today about sort of evolutionary perspectives on treatment and adaptive therapy. But I wanted to sort of go back to the basics a bit, since this is a tutorial, and sort of think about sort of this first question about just how do we get this evolutionary or phylogenetic tree from the type of data that we now obtain from tumors. So in a sort of simplistic view, we can take this DNA sequencing data, we can build out for each cell the mutations that we measure and whether or not we measure that somatic mutation in that individual cell. And this data is exactly what is input, sometimes called like a mutation matrix or a character matrix. This is exactly what's into the input for many algorithms that have been developed over the past few decades to build evolutionary trees or phylogenetic trees. And so the basic problem is, given this character or mutation matrix, here I've represented each mutation by either mutated or not, 0 unmutated, 1 mutated. How do we build this tree? And this tree has both a topology, which describes this underlying ancestor relationships, and then each branch also has a length, which is related to either the mutational time or actually the real time of that lineage. So this is a classic problem, again, many decades of work. And to skip a lot of stuff, I'll just say that sort of the most rich model is to do this with a probabilistic model. So we first make this assumption, we're going to come back to this, but the standard assumption in most phylogenetic reconstructions is that each character, so each mutation, evolves independently of the others. And so we can describe this tree, what's the probability of generating this observed data given the tree and branch lengths, as just the joint probability over each column of the tree, each mutation independently. And then for each mutation or character, we have some transition probability matrix that describes how we go from the ancestral state to a descendant state, in this case, zeros or ones, if I'm doing just this two-state model, in a certain length of time given by this branch length. And if we do this in the usual way, we sort of assume that this is a continuous time homogeneous Markov process. So it only depends on the length of this edge. It's parameterized by this rate matrix, which describes the transition probabilities between the states. All right, so in this model, there are things that are easy to compute and there are things that are hard to compute. So easy to compute is just given the tree and the branch lengths, I can compute this likelihood probability of the observed data. You do that column by column. So that's easy. There are great algorithms for doing that. Slightly more complicated is, given the tree, I can try to find and fit the branch lengths that best describe the data. And that's done using an optimization procedure, something like expectation maximization in the probabilistic setting. It's a non-convex optimization problem, but it's still something that can be done reasonably well. And then up to the hardest case, where if I just give you the observed data and you have to infer everything, what's the tree topology, what are the branch lengths, that's a hard problem. That involves heuristic search over the space of trees. And there's no good way to do that in this model except heuristics. You do Markov chain Monte Carlo, you do simulated annealing, you do choose your favorite heuristic for solving this hard optimization problem over this discrete space of trees. But people do it a lot, and there are many, many good heuristics, right? But
I just want to ask a dumb question
because
I don't want to get lost at the beginning.
Yes.
Okay, so just the assumptions of this model are that the different mutations are independent, but they have different probabilities than each other, right? That's the focus.
Yeah.
That is not, that mutation isn't completely random, right?
No. You have a rate matrix that parameterizes the probability of a state.
Why is it a matrix rather than just a vector?
Oh, sorry, for each state. So I have to describe, and maybe I should have written this down. Like I need the probability that 0 goes to 1 or 0 stays to 0.
Oh,
right?
okay.
So it's just
it's a skinny one.
Yeah,
okay.
yeah.
All right, and this independence, right? Could one of the early mutations be something that will affect the error correction?
Yeah, so there could be interactions between mutations, and those get hard to model.
Okay, so we're ignoring that possibility.
For now, yeah, I'm just setting up. This is hard. This is already hard enough, and then you can start making it harder in lots of different ways. And that's actually what I want to describe some of, how it gets harder in the cancer setting. Yes?
Yep, we're ready for you.
I was just wondering if the mutation probabilities are somehow estimable, or is that open as well? Like, I guess different mutations would have different probabilities.
Yes, this is a great question. I meant to put in some slides on this, but there was already too much else in this tutorial. So classically, in species evolution, there are models that are used to describe substitutions between the characters of DNA, so A, C, T's, and G's, and we describe like what these mutation probabilities should be. In the cancer setting, it's actually much more interesting and challenging to describe what these mutation probabilities are, because there's various somatic mutation processes that change the probabilities of different mutations happening. And they're related to things like the DNA repair process. And so there's a whole series of work, and I could have put in a whole tutorial on what's called mutation processes and how people estimate these mutation processes from data. It's actually a rich and interesting area. Yeah?
Sorry, so if you pick only one mutation, right? So you do it for one eye, then you only get these relatively simple trees that what you have on the figure.
Yeah.
Okay, but that's still very hard to...
Yeah, I mean, for one mutation, I mean, if you have one mutation and you only have two states, mutated or not, you're only going to be able to split the cells into two groups. So it's not going to be a very interesting tree. So what you do is you do this for many mutations, but it's just that when you're doing the calculation of the data, it's the joint probability over each of the columns under this independence.
But when you take the max at the end, do you have a product there also, or something?
Oh, yes, sorry. Yes, you're exactly right. I should have that. This is the calculation for one. We can do this. Here we need to max over all the mutations. And I'm sorry, this also should be maxed over all the mutations. So this should say M and M. Yeah, thanks. These were new slides, so a few errors. Okay, so.
And by the way, just in our brains, how big trees should we be thinking of?
Okay, so we'll get to that, and it has a lot to do with the data. So we're gonna talk about some of the data and some of the challenges, and that's actually where we're gonna go. So all that's been worked out in species evolution. There's a whole field, very rich, deep field. Many people still work in this area and deal with these issues. What's different in cancer evolution? All right, so the first major difference is really kind of the technology and the measurement, and it's actually an important difference. And so I wanted to start with this, and that's that our unit of evolution is the single cell. That's the correspondence to the species, if we were thinking about species evolution. But the measurements that we make are often not single-cell DNA sequencing of whole genomes that allow us to measure these mutations. So this is a very hard thing to do, to measure mutations in single cells reliably. And often then what we're trying to do is construct this phylogeny under some uncertainty in the measurements, right? So that's the first challenge, and we're gonna talk about that one in a few minutes. And then the next one is to think about, well, how is the genome changing? And cancer genomes are very complicated. So they have mutations that range from single nucleotide substitutions in the DNA sequence all the way through deletions or duplications of segments of the genome, so-called copy number aberrations, to various structural rearrangements. You can have a part of the genome cut out and invert. You can translocate between different chromosomes, all the way to really dramatic events like the whole genome in a cell can duplicate. This is called whole genome duplication. And so the range of mutations goes across these long genomic scales. And so we need to think about what that means for how we try to build these trees, which mutations we might use to be able to build these trees reliably. Okay? So this often relies on some specialized models or distances for evolution. So that's another challenge in cancer evolution that kind of distinguishes it from species evolution. And then a third one, and this will be the second half of this tutorial, is that there's a lot of interesting, and we heard about this yesterday, spatial aspects to tumor evolution. One on sort of the global scale is one I already mentioned, where cells leave the primary tumor and seed metastases at different sites. This is a form of spatial on sort of a macro scale. And then there's sort of the micro scale of individual cells within one tumor mass growing and dividing and mutating and migrating. Right? So there's the spatial aspects that we'd also like to model. In phylogenetic terms, they sometimes call this phylogeography when they're thinking about, say, species migrating between disparate islands or something in physical space. Okay, so for the tutorial, though we've been already going for a while, so the first half, the rest of this talk, I'm going to talk a little bit about some of the challenges when you construct phylogenies from actually the data we get, bulk and single cell sequencing. And then in the second half, we're going to talk about these spatial aspects with metastasis and spatial tumor evolution. Okay, so what is the data that we do get instead of getting this very reliable single cell sequencing data? There are roughly 3 technologies that have been used to measure tumor samples, measure mutations in tumor samples, so measure DNA. The sort of workhorse, and still a lot of the data we get, is from a bulk tissue sample. So you pull out a piece of this tumor, you sequence thousands of millions of cells together at once. So you get this aggregate or mixed measurement. Okay, so that's going to be something that's going to challenge this algorithm for building the phylogenies. More recently, there are these technologies that allow the tagging of individual cells with these barcodes and then sequencing them in parallel. So sequencing thousands of cells in parallel, but with some limitations that I'm going to describe. And in the second half, we're going to just talk about some newer technologies for actually barcoding individual spatial locations in a 2D tissue or tumor slice and sequencing what is at each physical location, keeping track of the location. So these spatial sequencing technologies. All right, so bulk. So most of the data that is published and that you see with mutations in tumors is from what we could call bulk tumor sequencing. So what is that? So we take a tumor sample, we could chop it up physically into a couple of pieces if we want, and people have done this, and then you throw it on one of these high throughput sequencing machines like Illumina, if you've heard of that, or PacBio more recently. But all of these give you a very fragmented view of your genome, and it's the genomes of all the cells in that sample together. So you get billions of these little short pieces. Each piece is a fragment of DNA from one of the cells. I don't know which cell it came from. And on some of these pieces, there's going to be these little somatic mutations here indicated by the triangle. How do I detect those somatic mutations? Well, I align these reads back to the reference genome sequence, and then I see where these individual differences are in the DNA sequence. And then I can count how many reads aligned to this part of the genome, how many of the reads have the mutation compared to how many that don't. And this gives me a proportion or a frequency, sometimes called the variant allele frequency. And if I have enough reads, and if the sampling process is uniform in the right way, I could think that this frequency I measured is somehow proportional to the fraction of cells that had this mutation.
So,
okay?
What is the current technology for the size of the reads?
So the current technology, I mean, there's sort of two ways, and without going into too many details, there's like the workhorse is this used to be called short read sequencing, Illumina. It's nowadays about 250 in length.
Okay, thanks.
One more question. There are 4 letters,
Yeah.
so you can substitute in 3 different ways. How do you?
Exactly, yes. So most of the time, and we're going to come to this in a second, somatic mutation is distinguishing at a particular position in the genome. There are 2 copies, one from the mother and one from the father at each position in the genome. We tend to observe only a single letter change at a position in the genome, somatic mutations, for the type of data we get. It's kind of rare that you get multiple different mutations at the same position. So usually we only see one of them, and we can characterize.
Can I?
Yeah.
So presumably a lot of the mutations are not going to be functional in some sense,
Yeah.
like no-code ingredients.
Yeah, exactly.
So that's going to affect our understanding of evolution, because if they're neutral, they're not going to do anything.
Okay, yeah, great question. If we want to build the tree, we don't care if they're neutral evolutions or driver mutations. They're all just characters of lineage. So the tree is about lineage.
Only about lineage.
Yes,
I
yeah. So they're all equally good as long as they're real. Yeah. Okay, so now instead of getting that matrix that was zeros and ones, whether each mutation is present in each cell, we now have these frequencies, values between 0 and 1. And this gives us actually a really interesting twist on phylogenetic inference. I call this phylogenetic deconvolution, where you measure instead frequencies of mutations, and you'd like to then recover the individual groups of cells, group them by mutations, while simultaneously inferring their history back in time. Okay, so by analogy, traditional phylogeny, species evolution, you measure whether or not a mutation is present in each of your species. The species are your leaves of the tree. You don't know the tree, so you go build it. I've already told you that's a hard problem. In this problem, we've sort of mixed the species together. So it's like we measure all the species, say, on different continents and the frequencies that they have mutations. And then now we're supposed to build the tree just from these mixtures. So we don't even know what the leaf sequences are. We have mixtures of them. So this is an even harder problem. All right. And so how can we address this harder problem? We're going to come to that in the last few minutes here. But let me set up kind of the other one, because they're going to have a related idea. The newer thing is single-cell DNA sequencing. So you extract these single cells. Now this is where things get hard, because there's only one DNA molecule per cell. That's a very weak signal. So in order to sequence it, you usually need to amplify the DNA in that cell. And you do some biochemistry tricks, and that's a not perfect process. So when you amplify, you'd love to get back this matrix of mutations by cells, but you don't get it back. Instead, you get a whole bunch of errors and primarily a lot of missing data, because you fail to amplify in parts of the genome that you're interested in. So now you have this matrix. This matrix might have, for some technologies, up to 80% missing entries. A lot of missing data. Where does it come from? Not going into too many details, this amplification can lead to non-uniform coverage, so there's biases in how things amplify. So instead of getting reads aligning to your position of the mutation, you get no reads there, because you failed to amplify in a certain cell. When you amplify, you actually do this PCR reaction that itself can introduce errors, And so you just get this very error-prone matrix. So, though similar idea, now we can say, well, this is not just any matrix. It's a matrix of an evolutionary process related by a tree. So we can try to simultaneously build the tree and impute the missing data and correct the errors. And so this is what I'll call a phylogenetic imputation problem, right? So traditional phylogeny is here. You observe the matrix, you build the tree, already hard, but now we have these two interesting variations. One, we observe a very error-prone and missing data matrix, and we try to build the tree while imputing the missing data. The other one, we observe this aggregate matrix where we've grouped rows of the cells together, and we have to like pull them apart and also build the tree. So two interesting problems in phylogeny, sort of driven by this, these limitations in the technology. So how do we solve them? So there are many ways. I want to show you the way that has been used a lot in the cancer studies that you've probably seen to date. And the way that they do this, because I told you that already this problem is hard, even when you know the leaves. So the way we're going to do this is we're going to simplify this evolutionary model. Okay? So we're going to simplify it by using, in fact, the simplest phylogenetic model there is, so-called perfect phylogeny model. Okay? So what is this model? So this model has only 2 states per character. That's okay, actually. It's not a bad assumption. Mutated, it could be a somatic mutation or not. There are 4 characters in DNA, as you mentioned, but we rarely see, that it goes to, more than one of them at a particular position. So this one's okay. Here's where it's actually a very strong assumption. We're going to say that over the course of this whole evolutionary process, at a particular position, a cell has a somatic mutation at that position only once. That happens only once. So as cells are mutating, a somatic mutation might happen at position 356,000 on chromosome 1, and that's the only time position 356,000 is mutated. It's never going to happen in some other cell during this process. So this is a reasonably strong assumption. This is sometimes called the infinite sites assumption. This is a standard assumption actually in population genetics, where the idea is that the genome is very long, 3 billion nucleotides for the human genome, times 2. So, maybe there aren't that many mutations we're going to observe and measure, so the chance of there being a mutation more than once at the same position might be pretty small. We're going to assume it's 0 here, okay? So we make this assumption. Then we get actually a phylogeny that we can build, okay? So we're going to assume that the root, the ancestor, we know this is no somatic mutations, all zeros. We can enforce this. And then these changes happen only on one edge. So what this means is that every transition from the 0 to the 1 state for each of my mutations uniquely labels an edge. And then I can take this matrix and I can build the tree efficiently. In fact, just linear time in the data. So I can do the whole thing, okay? So I showed you the one extreme where it's really hard and you have this continuous time Markov process. This is the other extreme where it's really easy because I've constrained the process a lot. All right. Now it seems like, is that useful at all? I hope to maybe show you it is. Yes?
Kind of technical question. Is the germline genome sequences for each patient for the tumor?
Typically, yes. These days, they tend to do it. People sometimes try to get away from it. But because we need to figure out which mutations were somatic, happened after birth, and which were inherited, we usually sequence sometimes called the matched normal. We sequence the normal, some normal tissue or some normal blood if it's not a blood cancer, and try to find that normal. Because we need to distinguish those 2 states. Okay, so with this model, can now pose a sort of straightforward problem, kind of a fun problem that we worked on. So you measure this for bulk data, this frequency matrix, this frequency of each mutation in each sample. What I'd like to do is just factor that matrix, pull it apart into 2 pieces. One is the mutations for the individual cells or clones. This is the 0-1 matrix that we would have liked to have measured, but we didn't. And then the second matrix is just how many of each cell is present in each of these populations, each of my samples. Okay, so I've measured in each of these bulk. I took 3 samples from this tumor. I measured the frequencies of mutations in each of those 3 samples. How many of each of these cells was in each of those 3 samples? Product of those is my observed data. I factor it, and we can actually show that you can compute this problem, compute this tree. It has a very nice combinatorial structure. It turns out to be a spanning tree and a graph you can build directly from this matrix. I'm not going to describe the construction. You can go see this, but you take this frequency matrix. You can build this graph by looking at the frequencies of mutations and sort of partially ordering them based on their frequency, and then you can pull out a spanning tree in a very specific way, and that solves this problem. Although it turns out to be a hard problem. In computer science terms, it's an NP hard problem, so there's no efficient algorithm for this. So we solve it using this mathematical programming technique, integer linear programming. So just to sort of summarize here, we took the simplest phylogenetic model, perfect phylogeny, we mixed it together, and we got actually a very hard problem already. So this says if I were to mix these other hard evolutionary models, I'm going to also obtain very hard problems.
Now, typical graphs have lots of spanning trees.
Yes.
So this thing here will...
Yeah, it's a spanning tree, and I didn't write all this down. There's certain constraints on the spanning tree.
So it ends up being unique when it exists.
No, and in fact, there's some non-uniqueness as well, which also becomes an issue. Depends on how many mutations you have, and but yeah. And this is my former postdoc, Mohammed Al-Kabir. He's now faculty at UIUC. He's actually written a few papers on looking at the non-uniqueness of these solutions.
Sorry, I don't know much about it, but this decomposition of the original matrix is surely not unique. So how do you go from left to right?
Yeah, and that's, I think, related to this question. So there's not a unique decomposition. And so you'll get many phylogenies that are consistent with your mixed data.
Spanning tree.
Mm-hmm. Yeah, and this is this question. There could be multiple, all of them are spanning trees, but there could be many spanning trees in this graph, each one giving you one of these trees. An important thing to think about when you're analyzing some data and they've shown you a tree in a paper, that there could be many, many other trees that also equally well fit the data. So here's an example of this. This was a large study. We were not part of this. There's actually even larger studies. This is a renal cell carcinoma. They had a few dozen patients here. This group in the UK, Tracer X group, has done this for hundreds of renal cell carcinomas and lung adenocarcinomas. They do this, sequence multiple regions from the tumor, build out these trees, and they're showing various mutations that have occurred either early or late in this development. And there's dozens of such papers now. Okay, so what about the other one, where we do single cell sequencing? Now we have this imputation problem. We're going to apply the same trick. We're going to use this perfect phylogeny model, so a very simple model. And then what we're going to ask is, under this simple model, we know how to build these trees very efficiently. In fact, in certain cases, there is, without any redundant rows, there's actually a unique tree. There's a one-to-one correspondence between these matrices and trees, again, under this strong model. So what we could try to do is take our data and try to find somehow the closest perfect phylogeny matrix. Okay? So this is like using the fact that this matrix is related by a tree. If we assume it's this perfect phylogeny evolutionary model, we can find the closest. And that's, in fact, what people have done in various ways. Again, it's a hard problem. If I do this in a sort of combinatorial way, where I just try to do a simple error correction, so I can replace the question marks that I don't know either by zeros or ones, so I can fill them in. I can maybe flip some zeros and ones to model errors. If I try to do this in a minimal way, it's also a hard problem. I can then do this in a probabilistic way, and this is what's been done by a number of groups. Here, if you have single cell data, you tend to have maybe hundreds now, thousands of cells. Each of these cells is kind of voting for a tree. You're not going to get single cell resolution. You're going to get a tree on groups of cells. And so you can do this in a probabilistic way, where you try to just compute the probability of any given tree given the data, and you end up sort of marginalizing over these cells. So you kind of sum the probabilities over groups of cells. Yes?
Yeah, I'm a little confused. So there's some set of perfect phylogeny matrices, and we want to find the distance in this metric, sort of Hamming type metric.
Yeah.
But there's many, so what is the definition of, again, of a perfect phylogeny tree that is used for this?
It's exactly this one. So, you know, it's a matrix that evolves according to this model. So it's a binary matrix. It has, you know, each character stating only once. I didn't actually put the condition. Maybe I should have. There's actually just a condition I can check to decide.
Well, that's what I was asking to
matrix
understand it from the matrix. Yeah. But it's a relatively small set of matrices in the set of all binary.
It's a set of relatively small set of matrices in the set of all binary matrices. Yes, they're very special.
I see.
In fact, I can tell you exactly what it is. If you look at any pair of columns, you can have all 4 possible states in the rows. So here I have, what are the for pairs? 0, 1, 1, 0, 0, 0, 1, 1. And you can see in any pair, here I only observe 1, 1, and 0, 0 in that first pair of columns. Right? So you don't get all 4.
0.75 for each pair, and then some composition.
Exactly.
And then that distance can be efficiently computed, or this is what you were saying? That's
so the distance between
the closest thing,
finding the closest cannot efficiently be computed. So I can't find it's the one that's the closest under this flipping model. Also a hard problem.
Scaling and n obviously on the size of the.
Yeah. So we're almost done, right?
Yeah.
Or
No, I was going to ask the question. Not saying that you're almost done, but
okay.
you are almost done. Is there any help to have for the same patient, both bulk data and single cell?
Yes, yeah, exactly. And people do that now because they have different sort of error profiles.
Right,
yeah. So like with bulk data, you know, you're measuring the genome pretty reliably, but you're getting this mixed signal.
Right.
With single cell data, you're measuring each cell very sparsely. And so if you have both, you can you have kind of deconvolution and imputation together, and it's sort of a very nice thing to do.
This reminds me of these people that are doing deep sea, trying to find, it's very similar, right?
Yeah. Some of these problems actually, you mean deep sea like the?
No. That people that were going on the oceans.
Yeah, exactly.
Yeah.
Some of these problems are actually very correspond to metagenomics, it's sometimes called, right? Where you sequence an environmental sample. Microbiome sampling.
Same thing,
a bunch of viruses. Just to show you the one example, and I'll maybe just show 1 or 2 more slides if that's okay. But this is just an example of this, right? Here's the sort of raw data we get. This is 255 single cells from a leukemia, so mutation cells. You can see it's kind of noisy. This is from a few years ago. Now the data sets are bigger. And then when you sort of run this procedure, you're going to build the tree. You're going to error correct the matrix while you're building the tree. And when you do that, you get these nice sort of block structures, which are somewhat apparent here. The rows and columns have been permuted to show the sort of blockiness of this. But it's sort of interesting. And you can do this mixing and this imputation in other models. I won't talk about that. So the data we get either mixed or with lots of errors. So building trees is not the same as what you have to do in species evolution. There are these interesting variations, and they're actually quite interesting algorithmically, some sense mathematically, and there are still some open questions in this. Let me just, if I can, because this will be important for the second half, I'm going to skip most of this. But these evolutionary models, we've talked now almost exclusively about these single nucleotide mutations. Those have been our signal of evolution. Copy number aberrations, these big duplications or deletions, are quite common in tumors. And they're in some cases easier to measure because if you delete a whole chromosome, you see that very strongly when you try to align the reads to the genome, right? It's a very strong signal. So people instead sometimes try to use these copy number aberrations. We're going to see this in the second half, how we've done this. So this is like you delete a part of the genome, maybe a whole chromosome, maybe half a chromosome. You duplicate a part of the genome. And so from the sequencing data, you can align the reads. And when you do that, what you get is what's called a copy number profile. So there's a lot of analysis that I'm skipping here. But as mathematicians, we can maybe think of this in a simple way. After someone else does all this data analysis, what we end up with is across the genome, just how many copies are there of each part of the genome. Normal human should have 2 copies of every part of the genome, 1 chromosome from the mother, 1 from the father. If you look at almost any solid tumor, you will not see 2 copies of every part of the genome. This is actually real data. You'll see, and these colors are indicating the number of copies. And in fact, you're going to do this, and we're going to see this in the second half. There's actually two integers you're interested in: the number of copies of the maternal chromosome and the number of copies of the paternal chromosome. Right? We start with one of each, one maternal, one paternal. In the cancer, it might delete the paternal chromosome 3, and it might amplify the maternal chromosome 8. So these pairs of integers across the genome are a different representation of a cancer cell. And so you can actually try to build phylogenies from these things. This I'm going to skip, just tell you that now you have very different models for how you build phylogenies because you're trying to analyze these strings of integers. And you have now operations that will duplicate or delete segments of these integers. That's the operation. So it's different from a single letter going from A, C, T, G, and we can view each of those independently. Now there's all sorts of dependence because I'm taking a whole segment and increasing or decreasing the number of copies. And there's other interesting things because if I've already deleted both copies of a part of the chromosome, I can't bring it back. So if I have a duplication of this segment, this thing was already 0. I never get it back. It's gone, right? It's not going to come back. So it's actually a very interesting model. And we and others have worked on that, but I'm going to skip that. And for the second half, go to spatial stuff. So that's the end of the first half.